All right. So I'm gonna plug some tissue culture clones. I like to do this in front of the hood. Helps keep everything sterile. So we've been sterile the whole time up to this point. So we've got, I mean, the tray isn't super sterile, but it has been cleaned with chlorine and alcohol. Same with these little pots. Can't autoclave these; they will melt. But the soil, which is not soil, it's Promix HP with cocoa or Promix cocoa, whatever that stuff is. This is autoclaved. So any of the mycorrhizae that were previously in this are now dead because they've been autoclaved. Because these plants are very delicate, and we don't want them to, you know, get attacked by anything. And really, it's not the mycorrhizae I'm worried about. It's these bales are stored outside, so could be fungus gnat larvae or mites or thrips or who knows what. But that could be an issue. So we're gonna get started. So oops, knocked it down. We got gelinade right here. This is our first one. There we go. So these guys, oh, goodness me. There we go. So they get rooted in these little peat, or I don't know what these are actually. They're Jiffy pellets. I'm assuming peat, but we root them in these deli cups like this, or realistically it's closer to like this, so they don't fall over as much. We usually do like 3 or 4 per container. These are sterile. These plants came out of tubes, which are sterile. So those plants start off looking like, let's see, where's it good? Here's a good example. Cold trait. So we'll pull these out. Not right now, not today. We're transplanting these. This is another video, but these will get removed from the gel, cut at the base, just like a normal clone. We pull some of the leaves off, do some rooting gel at the bottom, and then plug them into those containers. And then they take longer to root than most. I mean, you can see these were put in quite a while ago, and we've got decent rootage, but not a lot. I mean, they're very small, so these can't be too wet, right? They got to be pretty dry, and they take a minute, but no worries. All right, who we got? Dr. Bamboo? I don't know how to pronounce it. I'm bad at that. But yeah, Cherry Soda, fire. Gym Crush, also fire. Gym Crush, super stinky. But you probably already knew that. Okay, so let's get to work. So, as I said, we've got our clean containers, our clean media. We've got our clean clones, and now I mean it's nothing too crazy. Like, I mean, you can see how little these are. What I am gonna do is trim off a little bit of this material, because these we don't need, these little leaves. Boom. There we go. So, super easy. I just do 2 fingers down the cocoa, peat, whatever this stuff is, up, and done. And then I'm just going to do that so I remember who's who. And I'm going to label these after the fact. And then I save the lids, because we can just re-autoclave these. Easy peasy. Alright. Next up is Zeclair, which this one is going to be interesting because this plant I assumed was dead. I was like, come on, there's no way that thing is going to root. And it did. So we'll find out. I don't have super high hopes, but it does have roots. It does have meristem cells in there. It's not like rotted or anything like that. It's just very unhealthy. we'll find out. Fingers crossed. You know, it'd be great if it lives. And if not, I have more. But, you know, Z-Clair is kind of tricky in tissue culture for whatever reason. It's not, you know, it's not a one-size-fits-all kind of a thing. Sometimes they just don't want to be good. They want to be annoying. Why that is, I don't know. Boom, Zonuts. Let's get to work on Zonuts. This is like one of my favorites. This is an Umami staple right here. And this third one doesn't look great, so we'll just throw that one away. But again, we'll just do a little, get rid of some of these not so hot leaves. And then I've got some kind of growth on this plug, but clearly it's not affecting anything really, so no big deal. Oh, damn it. Okay. So that's one, same thing. So we'll just pop off a few of these leaves because we don't need them, and then yeah, happy little healthy plant. The other way I like to root them is in these cool little mini containers. So these things are pretty dope. So I mean, you can see how small some of these clones are. They start out just very little, only a couple centimeters tall. And you know, they do take a while to root, but that's how it goes with tissue culture. They're not fast until they actually get roots. That's kind of the trick. So now, I mean, you can see how crazy this one's already got 3 tops. 4, well, no, 3 tops. But we're just gonna get rid of some of these lower leaves. This lower node that we don't need. And this thing's gonna blow up. Like, absolutely. It's just gonna crack off. And that's it. So we're done. All we gotta do now is we're gonna put a clone dome on top of here because we don't want these to dry out because they have been in, you know, 100% humidity up until now. And then we kind of acclimate them just like normal clones. You know, they'll get a root system, they'll start to grow, they'll look normal. They definitely have some accelerated growth patterns. I mean, the whole thing with tissue culture is we want to start clean and sterile from the beginning, right? So there's no, I mean, through the process of getting them in culture, they're cleaned of any bacteria, molds, fungus, bugs. The only thing that can sort of make it all the way through is viroids, like the hop latent viroid. But there's a method called meristem culture, which allows you to remove basically like the stem cells of the plant and grow that out, which I actually have some meristems in here, which you'll notice you can't see them, and that is because they are invisible to the naked eye. I have this like big fancy microscope that I use, and the actual meristem itself is, I mean, it's hard to describe, but it would fit, on the very tip of one of these blades, and it would just like the tiniest little shape that you could kind of barely make out. It's wild. So those will grow, and then eventually those will look something like that. They start to eventually look kind of like plants, and then we'll get more like this, where they look even more like plants, and then eventually like that, and then they get rooted, and then they look like this. So that's how that goes. Alright, well, I gotta get a dome on these guys and some labels, so we'll leave you to it. But thanks for tuning in to the first Umami live stream. I'm gonna try to do more of these, because apparently people want to watch this stuff, which if you can stand my monotone voice, then hey, more power to you. Or just put music on in the background. Yeah.